everyone, and good morning to everyone as well. My name is Sina Mashishi. I'm an intern with the HSRC hosted by the University of Venda. So today I'm going to present part of my work that I did for my master's program under the supervision of Prof. JPO Ogola and Prof. JJO Odiambo. So I'll be presenting under the topic Evaluation of Biofertilizers and Chickpea Cultivars on phosphatase activity and P concentration in the chickpea rhizosphere. Chickpea is an important legume that is rich in protein, carbohydrates, essential micronutrients, and vitamins. It is a leguminous cover crop that is recognized for its importance in the ecosystem for the conservation practices. Globally, chickpea cultivation spans about 14.56 million hectares, producing 15 million tons annually. Will I be able to control the slides myself?
I'll control. Just tell me to move, then I'll move. And
okay,
there's some noise in the background there, please.
In my background?
Yeah, I suspect so.
Yeah, I'm in the office. I couldn't find a space. It is produced in many countries due to the multiple uses it has as food, livestock feed, and soil fertility booster. The Limpopo province and the Mpumalanga province were the first in South Africa to introduce chickpea production through research studies. The experiments were aimed to improve local chickpea production and involved different chickpea varieties. Next. The studies assessed the suitable cropping season, increasing grain yield, water use efficiency, phosphorus fertilizer, and biochar application. The results of the experiments revealed a significant increase in the parameters mentioned above. Tangwana and Ogola reported a twofold increase in grain yield of chickpea, whereas Ogola 2013 revealed an increase in water use efficiency with pea application. Next. South African soils are largely acidic. You can see anything. So South African soils are largely acidic due to the accumulation of hydrogen and aluminium or the leaching of base cations according to classification. The poor nutrient status of soil poses a serious threat to sustainable agricultural practices. So the low soil P levels in the cropping fields explain why local researchers included P application in their chickpea research studies. So phosphorus plays a key role in photosynthesis, the metabolism of sugars, energy storage, transfer, and cell divisions, including cell enlargement and transfer of genetic information. Also, it promotes healthy root growth, promotes early shoot growth, speeds ground cover for erosion protection. It also enhances the quality of fruit, vegetable, and grain crops. It is vital for seed formation as well. So phosphorus is one of the 17 essential nutrients that plants need for growth and reproduction, and is considered one of the 3 major nutrients along with nitrogen and potassium. So to reduce phosphorus deficiency through fixation, phosphorus fertilizers are applied in large quantities. However, this method has been proven not to be environmentally friendly, and also it is costly, and that results into soil degradation. So now, comparing the synthetic fertilizers to biofertilizers. Biofertilizers, they comprise of inexpensive, environmentally friendly, and renewable sources of plant nutrients, and they have since gained popularity and importance in agriculture. So recent studies show that biofertilizer application significantly improved chickpea growth and yields. Also, there have been reports that biofertilizer had a significant effect on acid and alkaline phosphatases in the bulk soil, roots, and tissue of chickpea. Moreover, studies also demonstrated that biofertilizer optimized phosphatase activity in the rhizosphere. Next. Prof.
It's now freezing. I'm not sure why it's not moving. Now having a challenge here of moving the slides.
Yeah,
Okay,
yes.
I've reshared again. Yeah,
Yes, we can see it.
you may continue.
So now the rhizosphere is the narrow region of soil around the plant root that is influenced by several factors like the root exudates and the associated soil microorganisms. It is considered the most active region of the soil as it receives the nutrients from the plants and from the microorganisms that are present around the roots. So now, in terms of the phosphatases, it is reported that chickpea has high demand for phosphorus, and in order to meet up with its demand for phosphorus, the literature shows that legumes have the ability to solubilize phosphorus by forming cluster roots and excreting organic acid and phosphatases. So these phosphatases are extracellular enzymes that catalyze the hydrolysis of p ester bonds in inorganic p-containing substrates to release the inorganic p into a usable form by plants.
The same challenge again. Okay, you may continue.
So these phosphatases are produced in the rhizosphere by fungi, bacteria, and plant roots. They are greatly influenced by the P substrates, P deficiency, and the P demand by plants and microbes. Reportedly, P fertilizer application has shown to decrease APASE activity, but to increase the alkaline phosphatase activity. Although chickpea develop some mechanisms to make up for the phosphorus that is actually deficient, the amount of P that is contributed through these mechanisms alone does not meet the P demand. So rhizosphere engineering with biofertilizers can enhance agricultural productivity and ecological sustainability by improving the nutrient cycling and promoting plant growth. So the research questions for the studies are as follows. Does application of biofertilizers affect extracellular phosphatases in chickpea? Does application of biofertilizers affect inorganic P accumulation? And the last one, does the effect of biofertilizers on extracellular phosphatases and inorganic P accumulation vary with chickpea cultivars? So this is my experimental. These are my experimental sites tabulated below. We had location 1, which was the University of Venda, situated in Toyando. Location number 2 is University of Limpopo, which their farm is located in Sefargale. So the type of soil in Toyando, it's deep, well-drained clay soils, and then for Sefargale, it's lumicent soils. So So the experiment consisted of 6 biofertilizer levels and 2 chickpea cultivars that were subjected to a factorial design that was arranged in a randomized complete block design. So the biofertilizer levels, we had mycorrhiza, we had Calpac, we had Rhizobium inoculum, we had a combination of mycorrhiza plus Rhizobium inoculum, we had combination of Calpac and Rhizobium inoculum, then we had zero control plot. So this was my experimental design outline, where it shows what T1 stands for, T2, which is the biofertilizers combined with the cultivars. So the experiments were planted around April for Sefargale and Toyando on different dates, of course. So the seeds were planted manually in rows with a spacing of 30 centimeter by 10 for both inter and intra-row. All experiments were irrigated using sprinkler irrigation system immediately after planting to encourage germination, to also encourage emergence, and to encourage crop establishment. So the experiments were always weeded throughout to eliminate competition between the required crop or the desired crop and the weeds. So at 14 days after emergence, 3,000 ml of Calpac was applied per plot. However, for microroot and Rhizobium inoculum, they were applied at planting or at sowing. So 5 grams per hole of microroot was applied. And then for Rhizobium inoculum, the seeds were soaked in the Rhizobium inoculum, and then we had to plant the ones that were not inoculated first to avoid contamination. Then we later planted the ones that were inoculated. So 5 soil samples were collected for the initial soil sampling. So these samples were collected to analyze for phosphorus, organic carbon, pH, electric conductivity, soil texture, and exchangeable cations for both locations prior to planting. And for the analysis of acid and alkaline phosphatase activity and inorganic phosphorus, soil samples were collected at 50% flowering on each plot, and then acid and alkaline phosphatase activity was analyzed following the procedure of Tabatabai 1994. And for inorganic phosphorus, was analyzed following the diacitric method modified by Duplessis and Becker. The data that was collected, it was subjected to the variance of analysis using a two-way factorial test to test the treatment's effects on the measured parameters using the statistics 10 software. So the two keys honestly significant difference test was used to separate the means that were significantly different at the level of P is less or equals to 0.05. So this is a table showing the results of the initial physical and chemical properties of soils at both locations. So we can see that Toyando, in terms of texture, the soils are classified as clay. And then for Sefargale, sandy loam. And then the pH level for Toyando, it's 5.92, which is acidic. And then even for Sefargale, it's 6.7, which is slightly acidic. And then the level of P in our soil in terms of milligram per kg. In Toyando, we had 11.23, and then in Sefargale, we had 15.47. So the effect of biofertilizers on the acid and alkaline phosphatase activity in Sefargale for the 2019 season, it shows that biofertilizers had a significant effect on both parameters. So the brown one represents the acid phosphatases, and the baby blue represents the alkaline phosphatases. So from our treatments, we can see that the combination of K plus R gave the highest increase in our acid phosphatases, which was then followed by the combination of M plus R and R when we compare it to our control. So K and M, they show to have a significant differences. However, the significant differences between these two, they are it's very slim, as it's denoted by the mean separations. So in the second season, biofertilizers show to have decreased alkaline phosphatase activity, because from the graph we can see that control is showing a significant increase in alkaline phosphatase activity compared to our treatments. So there was no increase in terms of biofertilizers. But for the acid phosphatase, there was a significant increase in terms of our biofertilizer applications. And from the graph, you can see that the combination of K plus R resulted in a much higher increase compared to our control. So in Toyando, for our first season, biofertilizers significantly increased acid and alkaline phosphatase activity in the rhizosphere. With for the acid phosphatases, the combination of M plus R resulted in an increased phosphatase activity, acid phosphatase activity. Then it was followed by our rhizobium, inoculum, and calpec. However, rhizobium, inoculum, and calpec show to have similar performance in terms of increasing the acid phosphatases. Then for the alkaline phosphatases, biofertilizers also show a significant increase. And then we can see that rhizobium, inoculum had the highest significant increase, which was followed by the combination of K plus R and M plus R. However, the combination of K plus R and M plus R, they show no differences amongst themselves. But when we compare our biofertilizers to control, they show a significant increase. So the effect of biofertilizers in our second season in Toyando, the biofertilizers significantly affected both our parameters, and mycorrhiza is showing to have a higher significance, and also the combination of mycorrhiza plus rhizobium inoculum is showing to have a higher an increased activity of alkaline activity in the rhizosphere when we compare it to our control. However, when we compare it to other biofertilizers, such as the combination of K plus R and R alone, there's a slight differences between the biofertilizers as compared to control. So please note that I forgot to mention in the beginning on my materials and method that the inorganic phosphorus accumulation, it was only analyzed for only one season, which is the 2021 season, due to finances. So these are the results that we got, and we can see that our location, which is Gale, University of Limpopo, had greater inorganic phosphorus accumulation as compared to our University of Venda, our second our second location. So from these treatments, all the treatments have shown to have greater. However, the combination of mycorrhiza plus rhizobium inoculum and mycorrhiza sole, they show no significant differences when we compare them to our control. Only K plus R and R as sole and K as sole showed an increase in our inorganic phosphorus accumulation. So in Univen, there was a significant difference. However, our control was greater compared to the combination of M plus R and K alone. So the effect of cultivar on inorganic P accumulation in Gale showed to be significant. And from our graph, we can observe that oxygen 3 resulted in greater inorganic phosphorus accumulation when compared to oxygen 7. So in conclusion, we can conclude that biofertilizers significantly affected the activities of extracellular phosphatases. Rhizobium inoculation appeared to enhance the effect of Calpac and mycorrhiza on acid phosphatase activity in both soil types, suggesting a synergistic effect. Acid phosphatase activity appeared to be greater in low P, which are the sandy soils. Biofertilizers decrease outpace activity in the sandy soil, but there was no clear pattern within our clay soils. Efficacy of biofertilizers may be influenced by the soil type. So our cultivars also affected the levels of inorganic phosphorus despite having no effect on our acid and alkaline phosphatase activities.
Thank you, Sina. So, any questions for Sina? So it's a great presentation. So learned a lot.
Sina, this is a good presentation. Very interesting data sets. How did you collect the rhizosphere soil? You did only collecting the rhizosphere, or also looked at the non-rhizosphere soil?
I only collected from the rhizosphere of the plant only. I did not collect for the non-rhizosphere.
Okay.
The non-rhizosphere was only for the initial soil sampling prior to planting.
Okay. So,
Yes.
how did you collect rhizosphere in terms of you are uprooting the plants and then shaking the roots to collect it, or like what methodology you followed?
So I had to use a hand shovel.
Mm-hmm.
So I would just collect around the plant so it will form a bulb. Then after I'll have to remove just a bit on the surface. Then I will just shake the plant into the plastic bag, then just sell the soil sample. Then I'll put them in the cooler box
Mm-hmm.
with ice, then take them to the lab, and then I place them in the refrigerator until the analysis were conducted.
Thank you.
Sina, this is Sridhar. Yeah, it's a great presentation. You have a multi-location trial at two different locations. I am kind of like very curious about the soils. I know the Limpopo soils looks like sandy. The Venda soils are like clay soils. Um, the pH is kind of like both acidic. So I was kind of like curious, was there a problem with the moisture holding capacity in the sandy soils? I know you showed higher inorganic phosphorus. So I was also thinking about the water holding capacity, and is that a factor influencing in the plants?
I wouldn't say it had a factor because looking at the studies that were conducted prior to mine, which were checking the water use efficiency and everything, they found out the level of moisture within our sandy soils. It's not just sandy soils; it's sandy loam soil. So they have an ability to hold moisture. So there was, even if there was a difference, it wouldn't be as much that it would affect the experiment, considering that this experiment was also not rain fed. So whenever there was a need, they would just irrigate.
Okay. So in general, do you need to irrigate more in the Limpopo soils compared to Venda?
Yes, a little. It wouldn't be much, but then yes, there would be a need to irrigate more often that side than this side. And considering that this was a winter experiment, and then we receive little to none rainfall during winter, unlike in our summer season.
Okay, good. Time for more questions. If not, I have one more question. Can you tell about the biofertilizer? What you have? Is it a commercial biofertilizer or is it prepared on the campus? Is it a vermicompost?
The biofertilizers that I've used for my experiments are commercial. So I've used Calpec. We had to order it from Cape Town, from a company from Cape Town, including the microroot. It was ordered from Cape Town. Both of them were actually ordered from Cape Town.
Okay. And both has and you also know what is the phosphorus content in those biofertilizers as well.
No, I haven't checked. I only know that microroot, it consists of the AMF or arbuscular mycorrhizal fungi. But for the Calpec, I only know that it's made from the green algae, and then it consists of the cytokinins, the gibberellins, and then yeah. Other than that, I can't remember clearly. I'm very sorry.
I did. Yeah, no problem. Yeah, it's very interesting. And is it surface applied or is it incorporated into the soil?
So the microroot at planting, when I placed the seed within the hole, I applied 5 gram of microroot immediately at planting. So for the Calpec, I had to wait until they emerge. Then I would apply 3,000 mil per plot. So Calpec, it was foliar application.
Okay. Any more questions for Sina? Yeah, you can unmute and ask.
Sina, can you hear me?
Yes, Prof.
Is there any way you can explain? I noted that inorganic phosphorus was much less at Univent site compared to Cypherquill site. Any explanation for that?
I think because of it could be influenced by maybe our pH level as well. It plays a significant role in terms of the availability of our acid phosphatases and alkaline phosphatases. So, as much as there was no significance, I strongly believe that they played a role as well into the hydrolysis of this solubilization of this inorganic phosphorus. So the type of soil as well, if you could remember, our Toyando, it's clay soil, and when it's clay soil, it's easy. They easily fix our phosphorus. So, and the soil is packed together. So I believe that plays a role as well as to how much of this can be available in terms of also influenced by the microbial activity. However, I did not check for it. I did not measure it, but I believe it has to do with the microbial activity and the pH.
Okay, thank you.
Any other questions for Sina? Yeah. If not, I have one more question, Sina. Like, are you seeing like the after effect of this on the subsequent crops at both the locations?
So a friend of mine planted a sunflower where I was performing my experiment, And for the first season, the sunflower did well. So because when you conduct your experiments, some experiments you won't have to, you don't have to change the location. You have to plant on the same location. For the second season, the performance was very low in terms of growth. So I believe that it did play a role.
And the clay soils in Venda, is drainage a problem? Because sometimes if there's a lot of clay, you may be having some drainage issues. Do you have any of those?
I haven't seen any so far. So yeah, I don't think so.
If there's no more questions, then we thank Sina one more time. So thank you very much for the presentation. So it came out very well. I also recorded the presentation. I will share the recording of the last and this one soon after this. I will post it to Dr. Jude, and he can actually share it with everyone.
Thank you.
Thank you, guys.
Thank you, Dr. Sridhar. So which presentation is the next month?
Next one will be Daphne. Daphne is next month, and Daphne is going to present next month. So I will post the flyer. Yeah.
Okay. Thank you so much.
Yeah. Thank you.
Bye-bye.
Thank you.